next speaker is Dr. Grant McFetridge. He has a PhD in psychology and originally worked in electrical engineering R&D as a consultant and adjunct faculty in California. In 1998, he founded the Institute for the Study of Peak States, where he still co-directs the research. He's the author of many books, including Subcellular Psychobiology Diagnosis Handbook, which is a key part of the trauma therapy training, Silence the Voices, Peak States of Consciousness, Volumes 1 and Volume 2, and the Wholehearted Healing Manual. Today, Grant is going to talk about human prion diseases.
This talk today is about human prion disease. And it's an interesting talk for me because it was so unexpected. Not in the foggiest imagination that I've ever had would I be talking about prion diseases. It all came out of the work we were doing, and it was just a complete and utter surprise. So let's get into what prion is going on with prion diseases. So today we're going to cover the discovery of prion diseases, which is a fascinating story, and then we're going to talk about what we found, which was completely unexpected. Then we're going to cover some of the prion diseases that we're finding that aren't known in the literature, and finally the treatments that we've come up with. And for my normal students, you can go back to sleep now that you've covered all of the outline for today. Okay, so let's see, can I say that? Let's start with safety and liability. This stuff is presented for educational purposes only. Don't experiment on yourself with the material that you're learning because you don't want to win the Darwin Award. And we'll be talking about some of the problems that we encountered when we were exploring this material. Working with this stuff requires training, and as like I said here in the write-up, only idiots make rocket fuel in their kitchen. And finally, if you're interested in some of the safety issues that we've encountered over the years, both Peak States of Consciousness Volume 2 and the Subcellular Psychobiology Diagnosis Handbook cover this stuff extensively. Okay, so let's start with the background. So biologists don't know what prions are. So that's a pretty bold statement, and let's go into how that is possible. So starting with how all this was discovered in regular medicine, the question is, how do sick people in Papua New Guinea and sheep in England tie together? And it turns out that they do tie together. They tie together by a particular disease that Carlton, and I can't pronounce his last name, he got a 1976 Nobel Prize for figuring out. So what he saw a connection was, there was a connection between a disease called scrapie, and it's called that because the sheep or the goats, they scrape up against things and they lose their wool and then they eventually die. Well, it turns out that in in Papua New Guinea, there is something called kuru. Stop me if I didn't pronounce it correctly. It's a disease that, again, where people slowly get it and they start acting very strange and eventually they die. And that fellow who got the Nobel Prize figured out that the two diseases were linked. And so how were they linked? Well, if you look under a microscope, they look very similar. The brain material of the humans in New Guinea and the sheep had this spongy look, like little holes were being made inside of their brains. And the symptoms were kind of similar in that they'd have trouble walking, they'd have involuntary movements, coma, and then they'd die, whether you're human or sheep. So linking those two was what got him in the Nobel Prize. So that disease in general is called TSE, and excuse me if I'm, it turns out there's more than just one disease with TSE. If I go back to here, you can see there's a whole bunch of them. One of the ones that we know that normal people have heard of is mad cow disease. And that's the one that caused such a problem in England. And it turned out a lot of people, a number of people got mad cow disease from eating infected cows. It's in the order of hundreds, so it wasn't a huge problem. But one of the reasons it wasn't a huge problem is that they got rid of the cows so that the people who were eating cow meat didn't get the infection. So let's just add to that. How did the kuru come in? Well, it turns out that these people in New Guinea were eating their ancestors. It was a ceremonial sorts of thing where you get the quality of the loved ancestors and stuff. But the problem was that their infected ancestors would then infect them. And so a very similar kind of problem. Let's go on. Okay, so let's look at the TSE problem. They found that there were layers of plaque building up in the brain. Well, that was strange. They also found that this disease was infectious. So how did they do that? They actually would take little bits of sample from an infected animal and they would inject it into another animal. And sure enough, after a certain amount of time, the healthy animal would get sick. One of the weird things was that they couldn't decontaminate well. So they'd use their surgical tools and then they'd clean them in the normal techniques, and their new animals would get sick also, as if they'd never decontaminated at all. Another thing funny about this disease was it's possible that it could take a lot of years to show up. For example, I can't remember if it's Kuru, but one of them had cases where it took 50 years for the disease to show after infection. Another weird thing about this was that whatever was causing the infections looked like it was smaller than a virus, or at least as small as a virus. It didn't have RNA or DNA, and they couldn't see it in a microscope. Finally, one of the things that was a good thing is it didn't cross species barriers, usually mad cow disease being an exception. Okay, so here's this data just accumulating, and an MD in the US, Stanley Prion, I believe that's how it's pronounced, he came up in 1982 with the term prion. And he hypothesized that a prion was a protein, and that it was self-replicating and it was causing these diseases somehow. So I'm just going to repeat that because this is key to this understanding. He saw this plaque. He found that it was made up of glycoprotein. He said, this is a strange looking glycoprotein, similar to something that's found naturally in the body, but it's bent and distorted. Maybe it's self-replicating, and somehow it's causing the symptoms. By the way, I'm going to call it a prion, even though I think the normal usage is often prion. So just hang with me on that one. Okay, so in 1997, Prion got a Nobel Prize because of his prion hypothesis. Now, this was kind of an unusual Nobel Prize because it wasn't proved to be true. But I have to say, given the amount of shit that he had to go through to say, look, there's really something here we don't understand. It's not a virus. It's not anything we know. We need to label it as something different. I think it was probably well-deserved. But so from that, especially with the Nobel Prize, people just started to assume that prions were understood, and they started doing tests and running experiments with animals and such to prove this hypothesis that this prion, this protein, could reproduce itself. And unfortunately, it turned out that that idea couldn't be verified. And in fact, nor could they figure out how a protein like that could cause the damage that was being seen. But you wouldn't know that if you didn't actually follow the literature, because so many people just assume prions are well understood and blah, blah, blah. This is another example of where medicine is, how do I put this? Where people just assume that somebody knows. And this is a good case where it turned out that people don't know. Okay, so what can you take from this? Prions they're considered incurable. So if you got it, you're screwed. Whether you're a sheep or a person, it's a big problem. Next, that the Prion hypothesis failed. The tests to see if the protein could create itself just didn't work out. The next thing is prions were assumed to be just a small group of TSC diseases, one with neurodevelopment issues. So that caused people to not look at other things too much. And over the last, I don't know, 10 years or so, folks have started to say, well, you know, there's these plaque accumulations in some other disease. We're going to call them prionoids because we can't seem to make them infect others, but yet they look damn similar. Okay, so that's basically a summary of the existing thinking on prions. Okay, and if you're interested in references, highly recommend Shifts and Drifts in Prion Science. That's fairly recent, 2020. It kind of discusses these issues, and a couple of textbooks as shown on the screen. Okay, now let's start to go into what we've been doing at the Institute. So this is kind of a gross picture. And as I say, yes, it creeps us out too. So where do you think this lives? Okay. So what we found when we started fooling around with the primary cell, that's the big cell inside the body that seems to regulate stuff, is we were encountering these metallic bug-like microorganisms inside of there. Okay. Well, you couldn't just go to a textbook and find something that looked like a bug living inside of cells. And so we're going, what the hell? And so we started to look into this. What are we seeing? What are they doing? They're there, so how come nobody's talking about them in the textbooks? We also found, not only in the cells of people in the present, but we found that when we did regression, that we were finding the same bugs infecting people very early in development, like ovulation, conception, that kind of places. So in the present, we were seeing these bugs, and in the past, we were seeing these bugs come in from our parents, in this case, usually a mom. Okay, so what are some of the characteristics of these bugs? Well, they've got these metal exoskeletons. So yeah, they sort of look like bugs, but they're made out of like metal. And inside the metal, there's some sort of squishy bits, but essentially when you look at them, you're seeing a metal exoskeleton. And they look like there's a lot of different species, and they sort of have a whole range of body structures. Interestingly enough, they all smell like sulfur, like rotten eggs. Little suckers, some of them can really move, given the size in the cell, and there's a lot of them. So when we first started this, we thought, oh, well, you know, maybe one or two or something, but it turns out that a normal person is colonized by these suckers. There's a lot of different bug species inside of them. What else did we figure out? Well, there's no genes inside these bugs. And there's a huge range of sizes in the species. Different species have different sizes. We also came across something really weird. It turned out that we could focus on a bug and cause them to burst into flames. Now, how we're doing this, not quite sure. It looks like maybe we're concentrating oxygen on the bug, and it's flammable. But it was very weird, something to observe. And finally, and probably most importantly for this talk, is that these bugs were making glycogens. Now, some of these fluids, the glycogen fluids, were caustic, but interestingly enough, a person who was exposed, for whatever reason, the bugs were making enough of this fluid, they would experience a negative emotion inside themselves. The fluid itself was experienced as a negative emotion, and that could be sadness or fear or you name it. Okay, so let's go back to the Prion hypothesis. Now, say you had a garden, and you're finding your plants were getting ripped up, and you don't know why. And in the garden, right next to your plant, you find a pile of poop. And you're looking at that, and for some reason, you only look in the daytime, the streetlight hypothesis that Dr. Likegaard talked about. So you're only looking in the daytime, and these poos are appearing when you're not looking. And there they are sitting. And then a day later, you find another poo. And then a day after that, you find another poo. And you're hypothesizing, well, what could be creating these poos? And it's wrecking my little bushes, it's tearing up my bushes. And you scratch your chin and you say, well, maybe these poos are self-replicating. And that's sort of the Prion hypothesis. Instead of saying, well, perhaps these things are being made by something else. And in the picture, it's made by a really nasty cat who has a real attitude problem towards our plants. Okay. Now, having said this, I'm not disparaging the Prion hypothesis. I actually admire it greatly because it really focused on the fact that there's something totally unknown going here. It turned out that from our perspective now, they got off track. They got off track with thinking that the glycoproteins being made by these bugs was actually where the glycoproteins were coming, that the glycoproteins were making themselves, rather than realizing that there was something they'd never seen before making these glycoproteins. Okay, so as Dr. Liegard said earlier, measurements and models go together. If you don't have a way to see what you're looking at, you make up models that aren't correct. For example, until the invention of the microscope, people just couldn't have a model that there were disease organisms that could be passed around because they had no way to see them. So your modeling and your measurement tools have to go together. We just got lucky in that our psychological measuring tools was able to stumble across these things. But it doesn't mean that they're psychological. It's just something that you have to actually build special stuff to find. And if you don't know there's something to find, you're not going to build the special stuff. Okay, so I hope I'm clear about that. Like I said, huge respect for those research guys. They were on the right track. They just didn't go far enough. Okay, so where do these prions come from? Well, there's a lot of weird stuff about these prions. They survive in temperatures that are really extreme. I mean, there's one study said that you could roast a material above 600 degrees C and still it would be infectious. Okay, so these prions can survive in huge temperatures. And as you know from your biology text, usually organisms that have a certain characteristic, there's a reason for the characteristic. So what might this be? Okay, so I'm going to go on. We actually had some very interesting data about this. And the first was we were doing regression through ancestors, looking for where these infections first started. And it took us all the way back about 72,000 years ago when Mount Toba exploded. And it looked like the whole biosphere got coated in these prion organisms. Now, the problem with that observation is it's untestable. I mean, if you walked into a scientific conference and said, oh, we've done a regression and we see prion organisms showing up 72,000 years ago in our imaginations, that's not going to get you very far, or other than kicked out the door. So we had this data point, but we didn't know what to do with it. And then we kind of got lucky. I was living in Hawaii writing the subcellular psychobiology handbook. And one day the volcano had a burp. Now, it had been spewing sulfur out for a while, but this day, something weird came out. It smelled like something you'd smell if you were welding. And immediately thereafter, my health started to deteriorate. I started gaining weight, which I'd never done in my life. And my sex drive went down and I'm going, what the hell happened? I thought, maybe this is male menopause. When I regressed backwards to see what had occurred, I found that while I've been breathing that crap from the volcano, there were bug species accumulating in the back of my throat. And eventually they were able to get into my body. So we have the smoking gun, as it were, the smoking volcano that says, whatever these bugs are, their natural habitat seems to be magma. Okay. So how did they get into people? I'm not sure. And I certainly welcome other people to investigate that. Okay, so so now it's a hypothesis, of course, but we think that the bugs that we're seeing inside the primary cell and that are creating symptoms and prions, as described in the literature, that cause TSC are the same organisms. Do we have proof of this? No, but it'd be pretty damn unlikely that there's a whole species of pathogens that nobody knows about and another one that nobody knows about. So I suspect that it's looking very consistent that these that the prions that are infectious and these bugs that we're seeing in the primary cell are the same organism, and we're just going with that. If, as you see us discussing more recently, we're just calling them prions instead of bugs. Okay. Again, I want to emphasize, why hasn't modern medicine found these? Well, if you don't know there's something to find and you have to build something special to see it, one of the reasons being we think these things might burn up in oxygen atmospheres. You're not going to, you just won't know that it's there unless somebody says, I think there's something here that's causing the problems that we can't solve. Another thing about these bugs is there's a lot of different species. They make a lot of different kinds of glycoproteins. So not just one kind as with the TSE issues, but a whole bunch of them. And finally, as are known, currently you can eat these prions and get infected. You can get them from the environment, from the ground, for example. It's a problem with when you have sheep and cows. But it turns out from our work, you can also get them from your parents. You basically inherit them. Okay, how are we doing for time? Anyone? Supposedly I'm supposed to have a clock here, but I guess I don't.
Yeah, probably another 5 minutes is fine.
Okay, so I'll try and go faster.
Yeah.
So these bugs that we were finding inside the primary cell were causing a ton of different symptoms. So one of the symptoms was these bugs could rip the crap out of your membranes, and people would experience this as being very painful. Now, why is that important? So this is a sort of a psychomatic or phantom pain. It's important because we were having that effect by just looking at these bugs in the primary cell. These little things were like wild animals, and they'd react either by freezing, running, or attacking. And unfortunately, the attacking could be bloody painful. Well, it turned out that you didn't have to be able to see in the primary cell to do this. It turned out focusing your attention on particular emotional tones was like looking at the bugs, and they would react and cause pain. This is one of the reasons why we don't want people screwing around with this stuff, because you can hurt yourself. And it turns out people are hurting themselves all the time, just not knowing where the symptoms are coming from. Some of the bug species, the way it works is they use emotional tones to disguise themselves. If that emotional tone is similar to, say, one that a person you know, your mom, your dad, brother, sister, a friend, when you focus on that person, you're simultaneously focusing on the bugs, and the bugs don't like it. Okay, so I'm going to go on because I'm kind of running out of time. Different bug species have different sizes. They attack different structures in the body. They have, and they do different things to the person. Okay, so what we have seen are different kinds of bugs, and we've seen different effects. One of the creepier effects is that on the slide, I'm indicating 3 different species that actually have psychoactive effects. So they change your inner psyche in generally bad ways. The weirdest one is one that can give you amnesia. We discovered that by accident. It makes people kind of go into la la land and forget what had happened, and it's a permanent forgetting. Peak state bugs, they can actually rip a state out of your being, and then it doesn't return. So a lot of different effects with psychoactive bugs. Cancer, turns out that we've seen bugs involved with cancer, whether they're causal. We have some evidence that shows that some of them are causal. We've seen organ cancer. We've seen bugs filling up a person when they had metastasized cancer. Seen that several times. In my own case, I had skin cancer where I saw bugs inside that seemed to be creating the problems inside the cells. I think one of the weirdest ones was we saw a particular bug species attacking a very, very small structure that's normally found inside the nucleolus. and the person would try and respond to this bug attack by creating a lump. But the lump was in the body and not actually where the damage was occurring. In this particular case, there was liver cancer, and the eyes got all yellow, and they were on death's doorstep. And we were able to intervene briefly on this bug. We didn't know how to get rid of it, but we could slow it down, and their symptoms just basically vanished in short amount of time. Their yellow went away. I mean, we're talking hours. Shocked everybody. Unfortunately, we weren't able to figure out a cure for this, and the problem returned, and the person eventually died. Okay, another weird place we've seen bugs is inside of COVID. Now, one of my colleagues was the first one to notice this, and this actually, because we discovered this in the first year of the pandemic, we were quite concerned about the creation of vaccines, because one of the ways you can make vaccines is you take the pathogen and then you basically kill it, and then you inject it into people. The problem is that there were two pathogens here. One was a bug pathogen, a prion, and one was the virus. And what we were afraid of is that people would start injecting large numbers of these prions into people. Fortunately, with the mRNA vaccines, that didn't become a problem because they weren't recycling old viruses. Okay, so we've also seen where bugs cause a problem, and then you have a fungi or a bacteria or both on top of that. And atherosclerosis is a good example of that. We've also seen bugs inside of another pathogen, and type 1 diabetes has a disease that actually has a bug fungal symbiote causing problems. And we've also seen some bugs just basically have bad attitudes, and multiple sclerosis, we don't know if it's causal or not, but the bugs are actually eating the histones on the DNA inside the nucleus. Okay, so there's a lot of different chronic diseases that are created by these bugs, which we're calling prions now. A lot of them are actually inside of your system from when you're born, because your parents had it, and their parents had it, and their parents had it, and they're causing symptoms that we just consider normal in the population. And yeah, and some references if you're interested. Finally, treatments. Now, prion diseases, as I said, in the medical literature are considered incurable, but since I've been working on psychoimmunology, we just considered them just another species we had to get at, and it turned out that our techniques do work on prions. So how's that possible? I think I'm out of time. 1 minute left. Okay, a little bit more time. How's that possible? As some of the people have spoken so far, and we've said last year, there's basically two models. One model is basically the victim model of disease. That's the one that we mostly know of in our lives, where our immune systems collapse under the assault of whatever pathogen it is. This is the model that we actually work with here. And that's where we've got this beautiful immune system, great castle walls, and we see the pathogen and we go, oh, that looks really good. Let's bring it in. And everybody's all happy to bring it in. And then, of course, you have lots of problems. The question is, why are you stupid enough to bring it in? And that's where we made real breakthroughs in this work. Okay, so back to prions. It turned out that the approach that we're taking for pretty much any disease works on prions. The only weird part was that it turned out we had to do the metal exoskeleton and the chewy stuff inside with different treatments. So some more general work about the approach we're taking in general for PI. It's really fast. Symptoms go amazingly quickly. We're still working on ways to make it simple. So a lot of this stuff is just proof of principle, and we're doing it in research, but it isn't ready for prime time yet. And we're also finding some people just have some weird resistance to actually getting rid of their pathogens. Not everybody, but a lot of people, and we're working out ways to work around that. Finally, because all this sounds pretty crazy to a lot of people, we have pay for results. That means if it doesn't work, it's free. So what have you got to lose? Except your disease, of course. Again, in summary, we've got potential prion treatments. It's basically a revolution in medicine to know that there's actually organisms creating these prions, and a lot of smart people are out there. If they knew to look for this, they could probably come up with dozens of ways to interact with these prions to get rid of diseases. And that includes the development of new drugs. I mean, psychological treatments are great because they're cheap, but not everybody can use them. It'd be nice to just take a pill and then just you'd be done with it. So in summary, we have some textbooks if you're interested in over the years how we slowly work this stuff out. And I'd like to acknowledge the crew that I worked with on these prion disease issues, basically all over the world. They've spent endless amount of hours of volunteer time, unpaid volunteer time to work on these issues. And I think that's it. Shane, how are we doing for time?
Yeah, so one person commented that it was very bold of you to talk about bugs at the conference. And
yeah.
the questions have exploded. So it's definitely been an engaging topic. We are out of time at the moment, but it's only half an hour until all the presenters will be back together for the panel. So there's going to be plenty to continue this discussion after Kate's presentation.
Okay, I just want to say one last thing. The bugs that we're seeing inside the primary cells and cells in general aren't the ones in your garden. These things sort of look the same, but they're not the same organisms. These are completely unique organisms. I hope that was clear in my talk. Okay, thanks.
Great. Thanks, Grant.